All right, we'll be starting the webinar now. Welcome everybody. My name is Robert Nyman. I'm the Manager of Partnerships and Programs at the Ontario Water Consortium. I'd like to welcome you to the final session of our Innovation Showcase Fall Series in collaboration with the University of Windsor, entitled Restoring and Protecting Freshwater Ecosystems at the Great Lakes Institute for Environmental Research. Before we get started, I just want to say a few words about the Ontario Water Consortium itself. The Ontario Water Consortium is Ontario's only sector support organization dedicated solely to water innovation. We bring together researchers, companies, municipalities, and governments to drive collaborative R&D, pilot testing, and real-world technology adoption. We help bridge the gap between research and market-ready innovation. And while there are many innovation organizations across Canada, none are focused specifically and exclusively on water like OWC is. OWC's collaborative model delivers measurable results. Over the past decade, we've helped mobilize over $90 million in project funding, supported or created more than 350 industry jobs, and advanced nearly 80 water technology projects. We've partnered with more than 100 academic researchers and helped build infrastructure, including wastewater test beds in London and Guelph. We do this by connecting innovators with adopters and aligning the right funding, partnerships, and support to move solutions forward. Before we begin today's session, a quick note that although this is the last webinar of our fall series, we plan to continue our Innovation Showcase webinars likely in January 2026. So please watch out for announcements and registration details before the end of this year. All sessions are free to join. Please also note the QR code here you can use to subscribe to our OWC newsletter. Now, I'm very pleased to introduce Dr. Michael McKay, professor in the School of Environment and director of the Great Lakes Institute for Environmental Research at the University of Windsor, who will begin today's webinar. Dr. McKay will provide an overview of GLIER's mission, research achievements, and collaborative work across the Great Lakes region. I'll also be adding a GLIER fact sheet to the chat for those interested in learning more about the Institute's work. We will then hear from Trevor Pitcher, Dr. Trevor Pitcher, who will highlight restoration, ecology, and biodiversity conservation at the Freshwater Restoration Ecology Center. And finally, Dr. Chris Weisener, who will discuss environmental stress impacts on freshwater quality and microbiomes. Mike, the floor is yours.
Thank you, Robert. I will go ahead and share my screen. Oh. All right, you can see everything? Screen's looking the appropriate way?
Looking good.
Excellent. All right, well, thanks everyone. And again, thanks to Robert Rahim and the Ontario Water Consortium for this invitation to join this afternoon to highlight the Great Lakes Institute for Environmental Research at the university. So safeguarding healthy Great Lakes, our slogan speaks volumes. Our position as Canada's leader on the Great Lakes is backed by decades of high impact scholarship, graduate education, community engagement, and environmental advocacy and action. Here you're seeing a remote sensing image, European Space Agency Sentinel satellite taken in March 2019. This stunning image, capturing the entire Great Lakes Basin, essentially cloud-free, was taken as the ice was thawing in that winter. So 16,000 kilometers of shoreline. This surpasses the coastline of the Atlantic coast of the US as well as the Gulf coasts. 250,000 square kilometers of surface area. These lakes are an immense system in North America, arguably the greatest natural resource in the continent, and shared by two countries and multiple tribes, First Nations, and Métis communities. Canada accounts for nearly 25% of the world's surface freshwater supply, while the Great Lakes represent 80% of all blue water resources in North America. And I think we all realize that the large lakes are integral to Canada's identity and economy. Of the 15 largest lakes worldwide that account for over 80% of the world's blue water resources, 6 are located in Canada. Swimmable, drinkable, fishable. These are goals for clean water advocacy adopted by many organizations, including the Great Lakes Institute. And this fact sheet reinforces the importance of the Great Lakes in many of these respects. The system provides drinking water to roughly 40 million people, 1 in 10 Americans, 1 in 4 Canadians. They support vast biodiversity. With diverse terrestrial and aquatic ecosystems and over 3,500 species of plants and around 350 species of fish. Collectively, commercial, recreational, tribal fisheries are valued at over US $5 billion annually and support over 75,000 jobs. For thousands of years, the region's waterways and terrain have facilitated trade. Its rich trade and transportation links now extend to global markets. By almost any measure, the Great Lakes region is one of the world's most important economic centers with a population over 100 million people, over 51 million jobs, and a GDP in excess of US $6 trillion. In fact, if it were a country, it would possess the 3rd largest GDP in the world. And Windsor, Essex plays a pivotal role in this equation as more than half of US and Canadian border trade happens in this region, with the Ambassador Bridge accounting for 25% of the bilateral trade between Canada and the US. So GLIER is part of the university. We're University of Windsor. We're located on the Detroit River, adjacent to the Ambassador Bridge, where our scientists address complex environmental problems across conventional disciplinary boundaries, such as the effects of multiple stressors on large lakes and their watersheds. For nearly 40 years, GLIER has been a lead academic research institute in Great Lakes research in Canada. We were founded in 1981 to provide dedicated research and training to protect and conserve the Great Lakes, while generating knowledge and data to help manage these unique and diverse ecosystems. The Institute immediately made its mark in 1988 when its scientists discovered the chemical blob of perchloroethylene in the St. Clair River. And soon after that, a student intern at GLIER discovered the first invasive zebra mussels in North America in our backyard in Lake St. Clair. In 1991, the Institute transitioned to become what it is presently, the Great Lakes Institute for Environmental Research. And since its inception, GLIER scientists have helped to shape federal and binational policy aimed at restoring, protecting, and managing our Great Lakes. Our faculty is diverse, transcending traditional faculty lines and acknowledging that the challenges faced by the lakes cannot be addressed by any single discipline. It comes as no surprise to anyone familiar with the Great Lakes Basin that environmental concerns compromise the ecosystem services and benefits of the lakes and connecting rivers, and thus pose threats to economic resilience and growth. These environmental concerns are almost entirely the result of human influence. On the bottom side of this figure, you'll see population distribution defined by hydrology. The Great Lakes Basin is home to about 10% of the US population and roughly 30% of Canada's population. If we consider the entirety of the 8 Great Lakes states, this accounts for over 80 million people, roughly 1/4 of the US population. Likewise, land use represents a stressor on the system, as shown in the top panel. The reds and oranges on that schematic show land allocated to agricultural use consistent with the Great Lakes Basin representing about 25% of Canadian agricultural production and a little bit under 10% of US farm production. Canadians and Americans living in the Great Lakes Basin have come to expect access to safe, clean, and reliable drinking water, and likewise access to lakes and rivers for recreational use. However, the legacy of natural resource extraction and industrial use, together with new pressures on freshwater ecosystems such as invasive species, microplastics, other contaminants of emerging concern, and harmful algal blooms challenge our beneficial use of these resources. Major federal projects are amplifying GLIER's standing both nationally and internationally in recent years. Current high-profile projects are promoting the development of smart Great Lakes. So the Rayon project of the University of Windsor, part of the Global Water Futures Observatories, positioning Canada as a global leader in fisheries conservation and management through the application of genomics and eDNA technologies, GenFish, and you'll be hearing a little bit about that from Trevor Pitcher in a moment, and supporting graduate training and amplifying Indigenous perspectives in fisheries through the NSERC Create Fishcast network. These programs bring national recognition to the university and are reaffirming GLIER's position as a nation's leader in Great Lakes research. As I mentioned, you'll be hearing more about the GenFish network from Trevor Pitcher, and then Chris Weisener will follow up and share some highlights from two new federally funded projects supported by Natural Resources Canada and the Canada Water Agency. A new high-profile project that kicked off last year reflects how GLIER is transcending traditional disciplinary boundaries, promoting collaboration, entrepreneurship, innovation, and knowledge mobilization necessary to help solve the challenges most pressing to the Great Lakes region. While our strength continues to focus on environmental research related to the Great Lakes and their watersheds, the Institute has been breaking down silos in significant ways to expand its reach to promote an innovation ecosystem and economic growth in the region. And so partnering with colleagues across science, engineering, our business school, along with the WeSpark Health Institute at the University of Windsor, the University of Windsor's Cross-Border Institute, and partners at Guelph, Waterloo, York, and the University of Toronto, last year we launched Inspire, a $16 million investment from the Canada Biomedical Research Fund, led by GLIER, that aims to integrate biomanufacturing and health sector supply chains, cross-border trade and mobility, and robust and timely cross-border pathogen surveillance, including environmental surveillance, to instill resilience and build capacity in the biomanufacturing sector in Canada. Almost everything we do in the Great Lakes requires some element of binational coordination, and facilitating this, Canada and the US created the International Joint Commission, whose Great Lakes Regional Office is located in Windsor, because they recognize that each country is affected by each other's actions in lake and river systems along the border. The two countries cooperate to manage these waters and protect them from the benefit of everyone. And the IJC is guided by the Boundary Waters Treaty, signed by Canada and the United States in 1909. The treaty provides principles rather than prescriptive guidance for preventing and resolving disputes over waters shared between the two countries and settling other transboundary issues. So given our location and the experience of GLIER researchers working on both sides of the border, we're able to effectively forge strong cross-border alliances, especially related to our emphasis on the Great Lakes and connecting channels. A few of these I show in this slide. We co-host the biennial State of the Strait Conference, where indicators of ecological integrity of the Huron-Erie corridor take center stage. We are the only Canadian university member of CGLIER, the Cooperative Institute for Great Lakes Research, a partnership between NOAA, universities, non-governmental organizations, and businesses which aims to achieve environmental, economic, and social sustainability in the Great Lakes. We're also a member of the Great Lakes Center for Freshwaters and Human Health, a $5 million center funded by the NSF and the National Institutes for Environmental Health Science, working at the intersection of how water quality affects the lives of people living in the Great Lakes Basin. And we're home to the only cross-border United Nations regional center of expertise, partnering the University of Windsor and Wayne State University, and focused on U.S. Sustainable Development Goals, addressing climate change, economic and health disparities, and social inequality to create a more sustainable future for our two communities that share an international border. Just a couple of examples here, and this relates to some research I do, winter limnology in the Great Lakes. We depend strongly on these cross-border alliances, specifically with the joint operations of the Coast Guard through Operation Coal Shovel, partnering with both Canadian Coast Guard and U.S. Coast Guard to sample during the winter, which is essentially a blind spot in our understanding of the Great Lakes. And a really neat result that came of this last year, we were contacted in mid-March following this image, which was taken March 10th, 2025 of Lake Erie, contacted by NASA asking if we had any understanding of what was happening in the lake. What was the swirls they're seeing in Lake Erie as ice is receding? And it turned out our Coast Guard partners had sampled on that exact day. we had data showing those swirls were actually phytoplankton growing in the lake at this time of year and setting up the very productive ecosystem that is Lake Erie. Finally, I just want to mention this. This is the Great Lakes Connecting Channels Monitoring and Surveillance Network. University of Windsor is the most recent node on this Environment Climate Change Canada network, whereby water quality and suspended sediments are monitored. Surveillance data collected from the connecting channels through the Great Lakes network to determine baseline water quality status, long-term trends and spatial distributions, the effectiveness of management actions, and determine compliance with sediment quality objectives, and identify emerging issues within the basin. And with that, I will stop my part of the presentation. Thank you for your attention, and off to Trevor.
Yep, thanks, Mike. So just sharing my screen now, and I'll just be 1 second as usual with these things. All right, so there we go. So usual technology. So thanks for that, Mike. Great introduction, of course, to some of the work I'll be speaking to. So I'll be talking about the Freshwater Restoration Ecology Center, which is a facility under the umbrella of GLIER. And it's sort of a unique facility because it really focuses on Great Lakes issues, but also with a outreach component as well. So I'll just show you very briefly. It's located right on the Detroit River in the municipality of LaSalle, Ontario. So it's a municipality that's partnered with GLIER. And because of that, we sort of have a principled approach to make sure that we have community relevance as well as research relevance. So our focus for research includes water issues, but of course also focuses on freshwater biodiversity conservation. We do a lot of things like captive breeding of rare and endangered freshwater fishes. We, as I mentioned, do community relevance. We talk about flooding issues and road salt levels. And of course, we also do some outreach with community groups in LaSalle and the greater Essex community as well.
Trevor, I'm sorry to interrupt. I don't know that we're seeing your presentation.
Okay, no problem.
I don't think you're in presentation mode.
No problem. Let me just.
Yeah.
Is it now? How is it now?
I've seen you move now. I still see your PowerPoint screen, not just the
okay.
not the full screen.
Apologies for that. Let's see. How's this? Is that better? Is that better?
Just give me a moment here. I'm not seeing anything change right now.
Okay, no problem. I can fix anything. Bear with me 1 second. Always these technology issues when you have them.
Yeah.
Let's see. Entire screen, window, share. How's that? One more try. Last one. Better?
There we go.
No?
Now I can see your full screen now. Yeah. That's right.
Okay. Yep. So apologies for that. I'll be very brief. I kind of cut a couple slides while Mike was chatting at the end there. So very briefly to remind you where we were is that the primary focus, among other things, is freshwater biodiversity conservation. And that includes things like what you see on the screen now, which is probably the most critically endangered freshwater species in North America called red-side dace. And they're cohabitated in the Toronto area, among other places. And the reason I bring up red-side dace, and hopefully you can see the screen again, is that this relates to what Mike referred to, which is one of the big projects called GenFish, where we develop tools, specifically new tools for industry, including optimizing eDNA, which is environmental DNA, where you can take a water sample and identify fish in any body of water, or the fish health panel on the right-hand side, where we can look at gene expression as a measure of health in these animals as well. And so we're looking to partner with industry and partners over the next several years to roll out some of this innovation. To give you an idea, here's a student, one of our GLIER students, grabbing water samples during the winter. And so with those water samples, he filters them, as you might be aware of eDNA. But what we've done with the GenFish grant is optimize it to provide us very detailed assessment of not just detection, but we're working on assessing abundance of these rare and endangered animals. And what you can see on this screen is just a detection of either presence or absence of red-side dace in the greater Toronto area. The innovation here is that we can do in 2 or 3 days what takes consulting companies in Toronto 2 to 3 weeks. And so we're partnering or looking for more partners in the industrial space to optimize some of this work to save them time and money. The gene expression panel shown here is also very useful for a bunch of industrial partners and municipalities. In this example, we've recently tested the genetic panel on the effects of road salt on the endangered red-side dace. And the way it works is you take a non-invasive small clip from their gill, and you can ask, in quotes, the red-side dace whether or not they're healthy based on their gene expression. So what you see on the screen is a lot of different genes related to immune function and other important health markers. And so we can do that, and we're looking for partners for these kinds of rollout as well. Second thing we do at FREC, the Freshwater Restoration Ecology Center, is community relevance. So this is a local issue where we had lots of flooding near the river in the last few years. And so we work very closely with the municipality to roll out on the left side. You can see some of the sensors that we rolled out. And this helped the town essentially create a pumping station to essentially prevent flooding in real time. So this was a very unique situation where in a matter of 24 hours, we'd gone from no sensors to 13 sensors up to allow them to monitor the flooding situation in real time. And last but not least, I mentioned earlier that the GLIER is also very into outreach. We're very interested in engaging with the community. And so the Freshwater Restoration Ecology Center is open for tours. If anybody's interested, you can reach out to us. We do lots of different tours. This is actually a school group from one of our Indigenous partners, the Walpole Island First Nation. And we have tons of school children every year visit, as well as other community members like retirement groups, etc. Last but not least, we create resources for the freshwater system, including books for children on the left. You can see the website on the bottom, and on the right for teenagers. We went with video games to try to get them involved in watershed management. And so we've tried to engage them over the years in different ways. So I know that was a lot in a short amount of time, but if you're interested in checking out, our website is located at the top, and you can check out all the features of the Freshwater Restoration Ecology Center, and we're open for visits any time of the year. So with that, I'm going to pass off to my colleague, Chris. There you go, Chris, all yours.
All right, thanks. How's that? Can you see it full screen?
Looks good, Chris.
Okay, excellent. Yeah, thanks for this opportunity. I'm just going to share a few highlights. Mike also kind of alluded to some of these case studies, but my involvement at the Great Lakes is really to try and understand or connect environmental stressors and to observe their impacts on freshwater quality, but also the microbiomes. Those are our unseen heroes that usually reside at the sediment water interface, but they also control a lot of issues with respect to water quality, nutrient availability, and contaminant response. So as I said, part of my interest in microbiomes is looking at disruptions to natural biogeochemical cycles that we often take for granted. This could be, nitrogen metabolisms or sulfur metabolisms, or even the decomposition of potential hydrocarbons that may enter these basins. So it's really taking this bottom-up approach while trying to identify suitable reference conditions, but then separating that out and looking at gradients of change. So some of the projects that we've been looking at over the past few years have obviously been related to anthropogenic systems, anthropogenic influences. These can come from agriculture or from urban. So we're really trying now to tease some of those nuances out, especially in our part of the world in Essex County, where we see a very expanding, I'd say anthropogenic footprint with respect to greenhouse operations, but they often work side by side with surface-based farming activities too. But one of the primary things we're interested in is looking at disruptions to our stream systems, our river systems that are contributing to Lake Erie, and possible connections back to adverse nitrogen cycling or something like that related to different events. So the big piece that's been missing in all this is our understanding of seasonal variation, as well as the impacts of increasing storm intensity. So to do this, we've again developed some interesting toolkits, I would say. And this is kind of fed into my own research interests, looking at developing molecular-based approaches to understand these microbiomes, these microbiomes that we can't really see visibly with our eyes, but we can measure through using these genomic tools to look at taxonomic traits, community traits versus expression, or what we call expression or activity traits. And that's really looking at the functionality. How are these microbiomes controlling the function that's perhaps contributing to water quality disparity or improvement. So in this first case study, I just wanted to show you this. This was some work that we did with AFC, Agricultural Agri-Foods Canada, and we were looking at a tributary that was feeding Lake Erie. It's an agriculturally dominant system, and in this case, we just sort of capture along the transect differences in storm flow versus base flow events. And we can see in red the concentrations of nitrogen in the upper headwaters of this stream, mainly coming from tile drainage, with elevated phosphorus and nitrogen components. As we move down gradient towards the lake, we enter a few systems, a golf course as well as a wetland. But we can see even with the presence of a very small wetland, it effectively filters much of those nutrient gradients or those nutrient issues by the time it reaches the lake. We also paired this up with an understanding of functional genes that were entering these water systems during storm events. And we did find a really unique correlation with respect to very specific gene markers, which were entering through the tile drainage at the upper part of the water system or watershed, and basically moving their way down that watershed towards the lake. And this is really significant when we start thinking about processes that are oxidizing ammonia to nitrate and producing excess nitrates. Another study that we did, again, very, very parallel with this. Now, this is down near Leamington, Sturgeon Creek watershed. Again, this is a watershed that's under pressure, under stress from multiple sources, both urban, agriculture, as well as expanding greenhouse sector. Was again to look at that through a cursory lens, just to sort of see what is actually happening with nutrient dynamics in that system. In this particular study, my PhD student looked at this over the summer months, over 3 consecutive years. So we had a really good, clear shot of the seasonal sort of comparators. And what we found was that over time, there were these pinch points, or these which sort of correlated with some of the urban centers along that transect. We saw excess high degrees of nitrate that were building up prior to entering the wetlands, which are, again, south along Sturgeon Creek before it enters the lake. And we also saw that this correlated really well with some very specific microbial functionality, let's just say. And the good news in this was that by the time we looked at the functions that were actually impacting the lake, things had returned to normal, or what we would classify as normal, post-wetland. So again, it kind of drives home the importance of creating wetlands in some of these buffering environments. Now, this study kind of led to the success of us getting this large grant working with the Canadian Water Agency and the Ontario Greenhouse Vegetable Growers. This study started last year in 2024, and it was to fill the gaps that we had observed. We did not have a really robust seasonal information on this area or this landscape. So this project is now allowing us to fill in those holes. So we can basically, we're doing sort of a seasonal monthly sampling, high frequency event, both comparing storm and base flow conditions. And this really built off a sort of cooperative projects that we've had with the greenhouse sector, specifically looking at greenhouse pond management status, and exploring ways to provide better solutions to improving the water quality related to some of these greenhouse ponds, stormwater ponds. So looking at this, obviously, in order to ascertain where these sources are coming, because it's a very complex part of the world, especially when we include greenhouses and we include traditional farming and we include urban impacts, we need to develop reasonable tools to basically assess this. And that in this slide here, I'm sort of showing a distribution of nitrate isotopes that we can use to sort of help us fingerprint different water sources. At the top there, I've got a whole collection of greenhouse stormwater ponds. We can actually see new stormwater ponds signatures as well as legacy stormwater ponds. And the difference there is with those new stormwater ponds, they're basically a reflection of atmospheric rainwater, which is what they should be. And the legacy stormwater ponds do evolve over time towards something that looks more along a denitrification trend. So that's actually taking on a sort of a normal cycle. Now, what's interesting, adjacent to those sites, we also have municipal drain mixtures, which again take on a different sort of chemical characteristic of fertilizer and soil nitrogen sources. So again, sort of mirroring this agriculture or traditional agricultural impact. And of course, some of the other ponds are manure sort of septic conditions. So we really can get a really nice fingerprint from different sources of water that we're trying to target. And again, we can couple this with taxonomic indicators. And here I show you, I say sink source here. This is basically water ponds that were sort of sampled that were basically producing nutrients and acting as sinks for nutrients. And if we look at the DNA profiles from these ponds, it was about a collection of 25, we can actually see that the DNA markers don't really show us too much with respect to suitable bioindicators of choice. But if we change that and we look at the RNA characteristics from the same ponds, we actually see a very clear distinction between nutrient sources versus nutrient sink environments. So this is really helping us trying to identify key bioindicator markers for these kinds of processes. That sort of leads into this project goal. As I mentioned, we've got these 4 main sources of agricultural pollution or nutrient pollution, which we're trying to track. And what's really novel about this new project is that we're not only using these other chemical diagnostics and taxonomic indicators, but we're also developing key diagnostic viral and eRNA markers for both peppers, tomatoes, and pathogens. So now we'll be able to separate what those contributions are. Are they coming from the greenhouse sector? Are they coming from municipal sources? And so on. And hopefully along the way, this also leads us into best management practices. And we've already coupled with a few groups in this regard, and we're using these technologies to help advance those BMPs. Our project started, as I mentioned, in the fall 2024. It goes to 2028. So we're just starting this project. We've got a very ambitious collection of field sites, some high resolution, some for monitoring. The red are basically sites of agricultural controls that we're using, and the purple are a natural waterway that we're using for background controls. But we do sort of we are sort of collecting very, very interesting seasonal data now on baseflow events. And just to finish off, I'll just show you this really quickly. This is data from our first preliminary year, and basically showing seasonal trends. So we can see that for a lot of our locations in spring and winter, there's not too much happen. And unfortunately, I do have summer data, but I don't have it processed yet because we've been collecting it all summer. But when we compare the 2024 autumn to these spring winter events, we can certainly see quite disparate big differences with respect to nutrient loading. and specifically along the urban sections of Sturgeon Creek, as opposed to, say, the agricultural references, which are opposite to that on the left part of the screen. And then there's a nice sort of nest of greenhouses, greenhouse impacted areas, which basically shows some ammonia drivers in the fall. And this, again, could be related to decomposition occurring in the stormwater ponds that they're adjacent to some of these watersheds. So that was really quick, and I'll just like to say thank you for that, and I'll finish off with that. Okay.
Thank you very much, Chris and Trevor and Mike. We're now opening up the floor for questions. You have the opportunity to raise your hand and I can unmute your mic, or you can drop a question in the Q&A section, or of course you could use the chat as well. We'll just give everybody a few minutes to gather their thoughts.
And Robert,
please.
Can I just add something?
Sure,
and
absolutely.
I noticed we have a number of colleagues from the Ministry of the Environment, Conservation and Parks here today, and just want to mention we have worked in partnership with MECP on a number of occasions. Chris has led recently a project on Lake Ontario tributaries, working specifically with Jan Thomas, who I see has joined us today. We've also, we're a node on the wastewater surveillance group, the provincial consortium, which was led by MECP. So I just want to acknowledge support of MECP for a lot of the work that we've done as well.
Yeah, it's hard to fill in all those gaps within 10 minutes. So.
All right, once again, you can either pose your questions in the Q&A section, or you can raise your hand. Or of course, you can use the chat. Just wait a few minutes.
I think we delivered it too well today.
I was going to say it's a rather quiet group, so it'd be a first that nobody has any questions at all. Sometimes it could take people a moment to sort of type up their questions or gather their thoughts. We'll wait another minute or so, but if there really truly isn't any questions, then you've done an excellent job. Of course, we should also mention you're able to get, we have a question here, but you're able to get in touch with any of the speakers, of course, directly if you want, or you can get in touch with me at the Ontario Water Consortium. I can always connect you. And we have a question here from Sylvain Campbell. I'm just going to allow your mic here. And now you should be able to unmute yourself, Sylvain, and ask your question.
Hello, can you hear me?
Sure can.
So is this like the tools that you're providing are really, really interesting and I see a lot of potential, but is that something that municipality could say how, if they want to evaluate the performance of their stormwater pond, could they actually send you samples, or is that something that could be done?
Chris, that'd be directed
to
you.
I'm just thinking. I don't see why not, but you're right. Like a lot of the stuff that we're developing is still, I wouldn't say it's mainstream, but we're getting there, right? So we're still sort of in this realm of trying to understand what are these suitable biomarkers, what represents a good fingerprint. And I think that's what the CWA project will help us define. So it's coming, I'll say that. But we could certainly look at pond health. Yes, we have a really good understanding of what to track, put it that way. Will you get the answer in 24 hours like Trevor's eDNA approaches? Probably not. But give me maybe a few weeks, maybe I could.
But Chris,
but
you know,
think I'll-
you had.
yeah.
I was just going to say, Chris, you know, Chris led a Genome Canada proposal early in the pandemic, and it was their largest was LSARP, I think, their competition, and it was essentially, you know, related to the GenFish project, developing toolkits to identify in Chris's case steps in biogeochemical cycles. Look at functionality in those, and that would be, you know, with those kind of toolkits. I think some of those will be developed as part of the CWA funding. Those could then be used. That would then make this analysis more rapid and allow you to test for more functionality within a biogeochemical cycle. And Trevor, sorry, I cut you off.
No, fine. I'm just going to add to Chris's point is that beyond the specific question, Sylvain, is that we do at GLIER have service provisions in many of the labs. We have suites of labs that provide services like we do eDNA for the feds. We do it for consulting companies, the province, MECP. We do some of the GenFish work. You know, as tools are developed, we certainly have the technical staff here at GLIER to offer services. So we do a lot of industrial work and things like that. So broadly speaking, we certainly have capacity to obviously run the water samples, but obviously as Chris develops more of these great tools, more opportunities come available for services we provide at GLIER. So maybe what we can provide in the future, we'll just have Mike provide is just a list of the different services we already offer at GLIER. If anybody's interested, you can reach out and we can help with that.
Yeah, and adding to Trevor's point, you know, GLIER is basically the home to the university's central facility laboratories. So these are a series of service laboratories. They are linked to the website for GLIER. They're also part of the CFI Navigator website. Where researchers across the country can look for different services. So yeah, they do support internally research from our collaborators, but also external clients. And Trevor alluded to one which is actually quite unique in Canada. It's our environmental genomics facility, which has capacity for sequencing, for qPCR, metabarcoding, other elements of genomics work. So yeah, that's what I'll do, Trevor, is I'll put that link to our facilities in the chat right now.
Yeah, because I could see Ontario municipalities, associations being interested in your work, like, you know, like OWA or like WEAO, like the Municipal Wastewater Associations, to be really interested in your work.
Yeah, we I'll be honest, like we do track a lot of stormwater pond progression. I've done these things for like in the city of Windsor, and yeah, you can obviously, yeah, you can see trends like in their performance and how they relate. You know, whether or not they're tipping over into a, you know, sort of a nutrient producing condition or not. So those kinds of things are fairly easy to assess. So
Thank you.
Thank you very much for that question. We do have a question in the Q&A section from MECP. Pradeep Goel asks, or states, just wondering about the role of non-point sources compared to point sources of P losses in the Leamington area.
what was that question again?
The comment was from Pradeep Goel at the MECP, just wondering about the role of non-point sources compared to point sources of P losses in the Leamington area.
Non-point versus point. Yeah, so that it depends on how you want to define that, right? Like, so much of the tribs that we've looked at. So for example, if we're defining that point source as a tile drain, do we see a significant phosphorus loss at those tile drains? No. We do see lots of nitrogen. Are there, when you go more diffuse, talking about non-point, again, that's a little bit of more of a question mark, because at that point you're getting a contribution from those non-points from a bunch of multiple sectors in Leamington. So that in lies why we're doing the CWA study, is to actually pinpoint what those non-point sources are.
I mean,
We
you've
do
got also,
see-
Chris, like the leachate piles, the compost piles, leachate
Oh, yeah.
happening. You know, those could be potential big non-point sources as well.
Yeah, exactly. And the problem there is, again, to figure out, well, it's really tracking down where those actually, I would call it a compost pile, more of a point source for some of these properties. But you know how that is managed, every property is different. That's one thing that we've noticed. So it's a little bit more, it is definitely more complex. You've also got a lot of other potential point sources out there. There's mushroom farms. There's, you know, differences in fertilizer application and things like that. So,
Thanks, Chris. There's another question in the Q&A now from Janice Thomas, also from the MECP. She says, lots of interesting work going on at GLIER. The virus work for source tracking is an interesting concept. Looking forward to hearing more about that one in the future. And then there's a question here for you, Chris. She says, Chris, what is the source of the urban influence in Sturgeon Creek? Wastewater discharge or a non-point source? Wondering what makes the signatures look so different in that area.
so that's a great question, Janice. We don't have an answer yet because we know that there's anomalies, right? And this is the thing, like, who's contributing to those sources? So right now we're as we speak, we've got people deploying collectors where we're actually growing biofilms on a lot of these identified drains that we know of. So we're basically looking at some of those viral components, some of those pathogen components to see whether or not they're showing up. So unfortunately, I don't have any data to help answer you at this point, but that is ongoing. And with respect to the bio trackers, I will say that the industry has been really cooperative. They've actually provided us with standardized waters that they use in different operations. So we're going to be fingerprinting those waters, and we'll hopefully get a clear understanding of what that viral component, what that chemical signature, and what that microbiome signature looks like. But at this point, I honestly can't say what is what until we get that data. Talk to me next year. Invite me back. I'll give another update presentation. Yeah.
Sounds good, Chris. We'll definitely keep that in mind. Are there any further questions from the audience? We are past our time, but if there's a final question, you can raise your hand, put it in the Q&A. I think there's something that just came through. Again, from the MECP, lots of questions. Vince Pelleggi, are there provisions/plans to make preliminary results and data summaries of results available? Example, data depots for planning purposes, noting that publications may take time to become available.
I can respond to this. So Vince, I think increasingly agencies want to see transparency with data that's generated by university research teams. As far as the Canada Water Agency project that Chris was describing, that was certainly one of the points that they want us to address, and we had suggested that we will be submitting data to a repository such as the Environmental Data Initiative that can make those data available to the public. Hasn't been done yet, but certainly on the to-do list, and we hope to get at least this first year's worth of nutrient data out into a repository such as that. And now they've made it such that you can actually publish those data sets. You can get data object identifiers associated with them, so they act like publications. So there's more incentive, I think, for researchers to do that now as well.
Thanks, Mike. Right. Any final questions from anybody? Again, you can raise your hand or write them in the Q&A or use the chat if you really like to. We'll give everybody maybe another minute, and if not, conclude the webinar. Again, you can always get in touch with the Ontario Water Consortium, Robert at ontariowater.ca, and I can connect you with Mike, Chris, or Trevor, or you can connect with them directly, of course. I see Mike has posted a number of links around GLIER, so I'm sure all the contact information is in there as well. I think this is a natural end to the webinar. Thank you very much to GLIER, the University of Windsor, Mike, Chris, and Trevor for great presentations, for the audience for being here today. I think it was a great webinar. We look forward to hearing new updates in the future. Thank you very much, everybody.
Thanks, Robert.
Okay.
Thank you.
See you soon. Bye.
Bye.
Well done. Thank you. Bye-bye.